name is Chang, and I'm a third-year medical student at Duke NUS Graduate Medical School of Singapore. In our third year, we have to undertake a project, and I chose to work with Dr. Ling from the Department of Gastroenterology at the Singapore General Hospital. Together with his postdoc, Dr. Linda Wang, we studied the numbers and function of myeloid-derived suppressor cells in gastric cancer patients. Funding for this project was provided by the NMRC of Singapore. As we know, the immune system plays an integral role in the defense against neoplastic diseases. Immune effector cells typically destroy neoplastic cells even before they can grow. Therefore, successful cancers must develop mechanisms to subvert this anti-tumor immunity. Recruitment of immune suppressor cells represents one of the many mechanisms by cancers to undermine our immune system. One of the more recently discovered immune suppressor cells are the myeloid-derived suppressor cells. MDSCs are a group of immature myeloid cells originating from the bone marrow and can be found in tumor and the peripheral lymphoid organs of tumor-bearing mice. They are very well characterized in mice and can be broadly divided into the monocyte-like or granulocyte-like subsets. MDSCs have been shown to be potent suppressors of the innate and adaptive immune system. The phenotype of MDSCs in human cancers is heterogeneous, and both monocyte-like and granulocyte-like MDSCs have been found in the peripheral blood of cancer patients. While increased numbers of MDSCs were found in mice with gastric cancer, there have been no studies on the numbers and function of MDSCs in cancers of the human gastrointestinal tract. We analyzed the peripheral blood mononuclear cells of gastric cancer patients and found that they had a highly granular subset of cells not seen in healthy individuals. Upon further characterization of this subset of highly granular cells, we found that their phenotype was similar to that of human cancers such as the NSCLC. When we compared levels of this defined set of MDSCs in our gastric cancer patients versus healthy individuals, we found that gastric cancer patients had a higher percentage of MDSCs in their PBMC fraction. When we stratified our cancers into the early and late stage cancers, we found that late-stage cancers had a higher number of MDSCs. Having established that MDSCs are increased in gastric cancer patients, we proceeded to examine its functions. We first studied the proliferation of PBMCs using CFSE dilution. We found that PBMCs stimulated by plate-coated anti-CD3 and CD28 antibodies proliferated minimally. However, depletion of MDSCs from the PBMC noticeably improved proliferation of PBMCs. In a slightly different experimental setup, we isolated T cells and MDSCs from gastric cancer patients using magnetic cell sorters and studied the effect of MDSCs on the proliferation of T cells. We saw that MDSCs halved the proliferation of T cells in these co-culture assays. When we studied the production of interferon gamma, a cytokine central to anti-tumor immunity, we found that MDSCs suppress interferon gamma production from T cells. Having shown that MDSCs were suppressive in vivo, we were interested to find out whether MDSCs could suppress T lymphocytes in vivos. CD3 zeta, a marker of T cell activation, was shown to be decreased in CD8 cytotoxic T lymphocytes that were suppressed by MDSCs. We therefore examined CD3 zeta expression in gastric cancer patients and found that there was a statistically lower amount of CD3 zeta expression in gastric cancer patients versus that of healthy individuals. This therefore hints that MDSCs are suppressive in vivo. We then proceeded to examine the possible mechanisms by which MDSCs may suppress immune effector cells. MDSCs have been shown to suppress T cell activation by a number of ways, but most MDSCs have been shown to interfere with L-arginine metabolism via arginase 1 and INOS. Using Western blot analyses of cells sorted by magnetic bead isolation, we have looked at arginase 1 and INOS production. We found that CD40 negative, CD11b positive cells of early and late stage gastric cancer patients highly express arginase 1, but this was not seen in healthy donors. On the other hand, INOS was not seen in these cells of gastric cancer patients. As a doctor in training, I'm keen on studying the possible clinical applications of our project. The 5-year survival of stage 3 gastric cancer remains at 10 to 25%, while that of stage 4 is less than 5%. Since we have shown that MDSCs are accumulated especially in late-stage cancer, perhaps inhibition of MDSC accumulation may improve the survival of gastric cancer patients. As our project has only been ongoing for a year now, we do not have survivor data, but we'll definitely be looking at it in the future. Ultimately, I hope that our findings can be translated to the benefit of gastric cancer patients.